Hello everyone, welcome to our next little video on histology. Now in this video we're going to talk about the structure of connective tissues. So connective tissues are going to be the opposite of epithelial tissue. Remember, epithelial tissue was dominated by cells. Connective tissues are going to be dominated by matrix. So they're going to have cells and some nuclei, but really mostly what you're going to see is this matrix, which remember is the stuff between cells. So matrix has two basic components to it, what's called ground substance and then protein fibers. So ground substance is, the substance that the fibers and the cells are suspended in. And in connective tissues, ground substance can vary really dramatically. So in some connective tissues, it's a solid, like in bone. In some connective tissues, it's a liquid, like in blood. In some connective tissues, it's, some a gel-like substance. So ground substance, really varies, and it's what the other stuff is kind of suspended in. There are also fibers in the matrix. So you have three types of fibers: collagen fibers, elastic fibers, and reticular fibers. Collagen fibers are typically going to be in thick bundles. Collagen fibers stain either pink or purple. Elastic fibers are going to be in thin bundles, and elastic fibers typically stain black. Reticular fibers are also going to stain black, but reticular fibers tend to be thicker than elastic fibers. Elastic fibers tend to be long, they cross the entire field of view, whereas reticular fibers tend to be short and branched. Now, like you see here, when we go over our connective tissues, you have these sort of three basic types of connective tissues. Connective tissue proper, which will consist of loose connective tissues and dense connective tissues. The fluid connective tissues, which are blood and lymph. We're really only going to talk about blood. And then supporting connective tissues, I actually call something else. I call these supporting connective tissues solid matrix connective tissues, because both of these, cartilage and bone, are going to have a matrix that's actually solid. And that helps to give these two tissues some features that they have in common. So the first are the loose connective tissues. Loose connective tissues are dominated by ground substance or cells. And there are three types: areolar connective tissue, adipose connective tissue, and reticular connective tissue. Adipose is going to be kind of the exception to the connective tissue rule, where adipose is going to be dominated by cells, but this will really be the only one that's like that. So first is areolar connective tissue. In areolar connective tissue, you're going to have lots of white visible ground substance. Those will be the white spaces that you see every so often. But areolar connective tissue is going to be the only one where you're going to see collagen fiber bundles and elastic fiber bundles kind of together in the same tissue. So the collagen fiber bundles are the thick, pink, pasty fibers that you see kind of crisscrossing this slide. Collagen fibers tend to kind of look like they're in the background because of the way that they stain. The elastic fibers are going to be the thinner black ones that kind of look like hairs, crisscrossing this whole field of view here. There's also going to be lots of different cell types in areolar connective tissue. So when you do see cells, don't expect them to all kind of look the same. There's like 12 different cell types in areolar connective tissue, and we're not going over that at all. Really, the dead giveaway for areolar connective tissue is the combination of collagen and elastic fibers in the tissue. You're not really going to see it like this anywhere else. Next is adipose connective tissue. So adipose connective tissue is different because it's going to be dominated by cells, unlike basically all other connective tissues. And what you're going to see in adipose connective tissue is a bunch of adipocytes or fat cells. So you can see one of those cells here. You'll see its nucleus kind of pushed off to the side like this, and then the center of the cell is going to be clear. So the center of the cell is going to have a huge vesicle that's storing a bunch of fat that people typically call the lipid droplet. And it stores so much fat that it pushes the nucleus and all the other organelles to the edges so that you can hardly even see them. Really, mostly what you see when you see an adipocyte is just what looks like a clear circle. So it kind of looks like soap bubbles when you look at this tissue. This is your adipose tissue. The last loose connective tissue is reticular connective tissue. So you will see some white ground substance here. One of the things that kind of makes reticular connective tissue stand out a little bit is it has these polka dot nuclei, right? These perfectly circular little nuclei that are going to be all over the place here. But really what differentiates reticular connective tissue from all other tissues are the short, black, branching reticular fibers that you're going to see. So notice when you look in this tissue, you see these black fibers here. But notice that these black fibers don't look like the elastic fibers we saw before. Notice that this fiber doesn't crisscross the entire field of view here. It doesn't go all the way across. It goes a short distance and then either ends or branches. And that's basically true for all the black fibers that you're seeing here. If you look carefully as well, you can see that those reticular fibers are also a little bit thicker than the elastic fibers you were looking at in the image before. So next are going to be your dense connective tissues. Dense connective tissues are going to be completely dominated by fibers. There's going to be fibers everywhere. There's going to be almost no visible ground substance and very few visible nuclei. There are technically three types of dense connective tissues, but we're just going to look at two of them: dense irregular connective tissue and dense regular connective tissue. So dense irregular connective tissue, which is what you're seeing here, these dense connective tissues, the two that we're looking at, are both collagen fiber connective tissues. So they're always going to be either pink or purple. This particular slide, the collagen fibers stain purple, which you haven't seen. This is the first time you've seen that. Now, dense tells you that the collagen fibers are going to be just everywhere. Irregular tells you that these collagen fibers are going to be arranged in all directions. And that means all directions in 3 dimensions. So you're going to have some of these collagen fibers kind of oblique like this at an angle. Some of them up here, notice, are horizontal. Some of them are a little more vertical. But then you're going to have little regions like this one where it looks like little cylinders. So these would be 3-dimensional ones. These would be collagen fibers actually coming out of the screen at you. These are going to be collagen fibers arranged in all directions in 3 dimensions. Now you do see a lot of white space in this particular slide, right? And you might be tempted to say that that's, sorry about that. So you do see a lot of white spaces, and you might be tempted to think that that's ground substance, but it's actually not. The white spaces that you're seeing here is artifact. Remember, artifact is something that you're not going to see in the native tissue. It's something that's introduced when the tissue is prepared to be sliced and put under the microscope. So the reason you're seeing all this white space in this tissue in particular is because this is like a bunch of cylindrical bundles. Imagine like a bunch of sticks kind of bundled together in all directions. Now, one of the things they have to do when they prepare these slides is they have to dehydrate the tissue. They have to remove the water. And when you have like these cylindrical bundles and you remove the water from them, they shrivel up and they shrink away from each other, leaving these gaps that aren't normally there. Next is dense regular connective tissue. So dense, again, means tons of collagen fibers. And you can definitely see that here. This one, you don't see the same amount of artifact, right? There's not as much white space. There's a little bit of artifact here and here. But this really shows you what a dense connective tissue would look like, right? All fibers, very little of anything else. Now, dense regular means the collagen fibers are going to be arranged parallel to one another. So that's why this dense regular connective tissue kind of looks like hair, because all the fibers are going the same direction. And they're all parallel to one another. Next are going to be the solid matrix connective tissues, which are going to be cartilage and bone. Now, like I said, what these two connective tissues have in common is their matrix is going to be a solid. It's not going to be a liquid or a gel-like substance. It's going to be completely solid. So imagine if you're going to be one of these cells living in this tissue. This means you're going to live your life essentially embedded in concrete. If you're going to live your life like that, are you going to want to be right up against the walls that surround you, or are you going to want to have a little bit of space between yourself and the concrete that's around you? So you're probably going to want to have a little space, and these cells are no different. So when you look at the cells in cartilage and bone, you're always going to see a little white space between the cell and the solid matrix. That space is called a lacuna. Lacuna is singular. If we're talking about all of these, it's lacunae. You just add an E to the end. Okay? So in cartilage and bone, the hallmark is always going to be cells in lacunae, cells in these little spaces in the solid matrix. So we'll start with cartilage. There's three types of cartilage. The first type of cartilage is hyaline cartilage. This is the most abundant type of cartilage. When you look at hyaline cartilage has collagen fibers in its solid matrix, but the collagen fibers are not visible. Now, the reason that we mention them is the collagen fibers, even though you can't see the individual fibers, give the color to the matrix, to the solid matrix. So notice that you have a smooth pink to purple matrix because of the presence of those collagen fibers. Then you're going to see these cells. These are cartilage cells, so they're called chondrocytes. So you have chondrocytes in lacunae in your hyaline cartilage. It looks like this. Okay? The next type of cartilage is called elastic cartilage. So in elastic cartilage, you're still going to have cartilage cells called chondrocytes, and they're going to be in lacunae. But in elastic cartilage, these chondrocytes are really large. So you have these really big cells in these really large lacunae. That's one of the things that you can see with elastic cartilage. But really the dead giveaway is you see these chondrocytes and lacunae, but then when you look in the matrix, you see these black fibers. So these are elastic fibers. So the combination of chondrocytes and lacunae and elastic fibers in the matrix, that's elastic cartilage. Next is fibrocartilage, the last type of cartilage. Fibrocartilage also has a ton of collagen fibers in the matrix. They're going to be arranged parallel to one another, similar to what you saw in dense regular connective tissue. And so fibrocartilage is oftentimes confused for dense regular connective tissue. But if you have a good eye and you look carefully, you'll see an important difference. When you look at the cells in fibrocartilage, they're going to be really tiny, really small nuclei with really small lacunae around them, really small spaces around them. And that's really the dead giveaway that this is fibrocartilage and not dense regular, because you'd never see those lacunae in dense regular connective tissue. Now I know that takes a lot of practice. You guys aren't going to have time for that much practice. So instead, what I give you is sort of a different staining technique. So notice that the collagen fibers here are not pink or purple. They're actually more like teal. And that's because a different stain is used. That stain makes the collagen fibers teal, and it makes the nuclei stain red. And so if I were you, that's one of the things I would definitely look at. Look at the difference in staining here and let that help you. Realize, oh, this must be fibrocartilage because it's not, the pink purple staining that I'm seeing in all the other slides. Next is bone. Now, the bone that you're looking at here is from what are called ground bone slides, where they dry out a bone and then they grind it up into a bunch of little flakes and sprinkle it on the microscope slide. So in this kind of ground bone slide, you're not going to see any nuclei of any osteocytes or any bone cells because those cells are gone before the preparation occurs. So this bone comes from what's called compact bone. Compact bone is like solid bone tissue. And you guys will learn later that not all bone is compact bone. But that's what we're looking at here. And so what you're seeing in this slide is you should see a bunch of these dark spots. Those dark spots are the lacunae, where those osteocytes, those bone cells, would live if this were, still a living tissue. Now, this is compact bone, and you're also going to see these eye-like structures that look like little eyes. These are called osteons, and that's a hallmark of compact bone, and you can use that to sort of help you identify this as compact bone as well. Last are the fluid connective tissues, which remember I told you have a liquid ground substance, and the two examples are blood and lymph, but we're really only going to look at blood. We're not going to look at lymph at all. So this slide here is a blood slide. So what this slide is called a blood smear. So what you do here is you take a sample of blood and you smear it across the slide. Because remember, to prepare things to see under the light microscope, you have to slice them thin. Since blood is a liquid, you can't really slice it, right? You can't cut it with a knife. So what you have to do instead is smear it and thin it out. And so that's what you're seeing here. So inside this blood slide, what you're going to see are tons of these little pink donut-looking cells. These are your red blood cells or your erythrocytes. And one of the things you should notice about them is that when you see all these pink cells, notice that none of them have a nucleus. They're all anucleate, meaning they do not have a nucleus. And that's really unique. You're not going to see that in very many places. Okay? The other cells that you see in blood are going to have a purple nucleus. So that'll be just the 4 cells that you see with the purple nucleus. These are actually your white blood cells or leukocytes. White blood cells or leukocytes are the only nucleated cells in blood. Red blood cells are never nucleated. So if you see a nucleus, a purple nucleus, is a dead giveaway that you're looking at a white blood cell, not a red blood cell.